name is Simpson, and thank you all for being here. So as you know, global warming is becoming an increasingly more severe issue, and this leads to the exacerbation of many already existing issues, such as the melting of permafrost, inclement weather conditions, and the spread of disease. And a major contributor to global warming is greenhouse gas emissions, and this causes heat to be trapped within the Earth's atmosphere. And one major contributor to greenhouse gas emissions is production of petroleum as a form of gasoline. An alternative to petroleum is biofuels, and this is produced from renewable resources and natural resources and leads to a much lower greenhouse gas emission value. And one type of biofuel is bioethanol, which is produced as a byproduct of the fermentation process of microorganisms. Fermentation takes simple sugars such as glucose and fructose, and through a series of reactions converts those into bioethanol. And bioethanol sources come from a few different types of biofuels. One is first-generation biofuels, which comes from sugar, wheat, and corn. And whenever these complex sugars that are present in these food sources are broken down into monosaccharides or simple sugars, microorganisms can then ferment these into bioethanol. However, first-generation biofuels compete with the food industry, so that's why second-generation biofuels are being more sought after. And this includes food waste, such as apple pommes, sugar beet pulp, and the skins of peaches and oranges. And a polysaccharide that is present in high abundance in pectin-rich biomass is a polysaccharide called pectin. And pectin can be broken down by an enzyme, which is a pair of molecular scissors called pectinase, into glucose and fructose, which again are the small sugars that then microorganisms can ferment to make bioethanol. One microorganism that is capable of fermenting fructose and glucose to make bioethanol is Saccharomyces cerevisiae, which is a kind of yeast. And you all might be thinking, this seems like a really great alternative for bioethanol production. Why don't we use this more? And that is because yeasts are very stressed out whenever trying to produce bioethanol in industrial conditions. Industrial conditions include acidic pH, high temperatures, osmotic stress, and high ethanol concentrations. And there's not one ideal strain of yeast. You either have yeast that die in industrial conditions but then express the pectinase enzyme, or you have organisms that are very robust and they do not express the pectinase enzyme. So there are two options of going about genetically modifying yeast to try to get them to both express pectinase and survive in industrial conditions. Option one is what I investigated with my lab partner Derek in the summer of 2024, and we took an industrially weak strain called Saccharomyces cerevisiae 09448 and genetically modified it with UV mutagenesis and directed evolution to try to get it to better withstand industrial conditions. I'm going to save you all the burden of looking at a bunch of data that was insignificant and did not work, and so we looked into option two, which is mutating an industrially robust strain to try to increase expression of pectinase. And so I'm going to talk about option two, which is what I did in the spring and summer of 2025. Saccharomyces cerevisiae ATCC 201388 is a strain of yeast that has a gene called DLS1. And knocking out DLS1 in previous research has shown to increase the expression of the PGU1 gene, as well as increase expression of the pectinase enzyme. So what we investigated was using CRISPR-Cas9 to specifically knock out the DLS1 gene to hopefully increase pectinase expression in ATCC. CRISPR-Cas9 is a way to make targeted gene edits, and there are three required components for this mechanism to occur. One is the Cas9 protein, which acts as a pair of molecular scissors cutting your target gene. Number two is a pair of guide RNAs that complex with the Cas9 protein to direct it to the target gene site. And this acts as a map whenever it's transferred into the yeast. And third is a DNA repair template that acts as glue to repair the double-stranded DNA break that Cas9 breaks. And there is three steps to the CRISPR-Cas9 system, which is the recognition of the target sequence using the guide RNAs, the cleavage by the Cas9 protein, and the repair by the DNA patch. And so our plan was to take ATCC, which expresses the DLS1 gene, and introduce a CRISPR-Cas9 system, therefore knocking out DLS1 and hopefully increasing pectinase expression. Before we were able to do this, we needed to make sure that the yeast had the proper instructions to make the Cas9 protein, the guide RNAs, as well as the patch to seal that double-strand break. and so we had to introduce some plasmids into the yeast. Two of these plasmids are on the left, L201, which has some specific genes on it of importance. There's a green fluorescent protein gene, which makes any organism having this GFP to glow green. There's the actual Cas9 protein itself. There's a kanamycin resistance gene, which is a type of antibiotic resistance gene. And there is a leucine oxytrophy gene, which will be significant in later steps. Our other plasmid is YTK74, and this has a chlorine phenol resistance gene, which is another type of antibiotic resistance gene, and a uracil oxytrophy gene, which will also be significant in later steps. And so how did we do this? First, we had to isolate the L201 plasmid from a strain of E. coli that carried this plasmid using a mini-prep technique. And then we performed what's called a Golden Gate reaction so that we can get the guide RNAs into the L201 plasmid backbone. So how this works is we have a GFP gene, which again makes any organisms that possess this plasmid to glow green. And then what we did was add a restriction enzyme called ESP3I. ESP3I cuts on either side of the GFP gene, therefore removing the gene. And in its place, we added the DLS1 guide RNAs, and then we sealed the guide RNAs into the plasmid backbone using a ligase enzyme, which acts as glue. And after that, we have our modified L201 plasmid that has the DLS1 guide RNAs in place of the green fluorescent protein gene. Our next step was to transform this modified L201 plasmid into competent E. coli cells and make a bunch of copies of it so that we can eventually transform it into yeast and then select for the E. coli transformants. A transformation works, and I know that this is not a bacillus, which Dr. Edwards is very picky about, but we have an extracellular piece of DNA. The E. coli basically just eats the plasmid and then expresses the plasmid and whatever genes are on it. And how we selected for transformants was we had an LB agar plate that had kanamycin in it. And once again, the L201 plasmid has a kanamycin resistance gene. So if an organism expresses the plasmid, they'll survive. If they don't have the plasmid, they will die. Another way that we selected for the appropriate transformants was not all organisms were transformed with the modified L201 plasmid. Some still had the wild type plasmid. And the wild type plasmid holders will glow green versus the non-wild type will be white, and we selected for the white. This is the result of our plate. So this is an LB kanamycin plate. The two left quadrants have our two transformants, trans 1 and trans 2 with the modified L201 plasmid. The top right is showing a regular E. coli that does not have the L201 plasmid, and because of that has no kanamycin resistance and therefore was unable to grow. And the bottom right is our E. coli with the unmodified L201 plasmid, so it glows green. Our next step was to confirm that our plasmid had the guide RNAs and no GFP, and so we did a diagnostic restriction digest. And so we added a different restriction enzyme to the L201 gene that on the wild type plasmid cuts in 4 places, producing 4 different chunks of DNA. However, we removed the GFP gene, and therefore we removed another PVU2 restriction site. So whenever we add this restriction enzyme, we should only have 3 restriction sites and then 3 different chunks of DNA. The bottom 2 chunks that are the smallest are the same with the modified and unmodified plasmids, versus we have a really huge chunk as a result of the removal of the GFP gene. And so as a summary, we should expect 4 chunks of DNA for the modified or unmodified plasmid and then 3 chunks of DNA for the modified plasmid. And we run this on what's called a gel, which we can see how big each chunk of DNA is. On the left is a ladder that we add, and we know exactly how big each of those chunks of DNA are and the number of base pairs. In lane 1, we have the unmodified plasmid, and then lanes 2 and 3 have trans 1 and trans 2's plasmids, respectively. And we have the 2 bands that should be the same across all 3 of the samples, which we can see. I do want to point out that I did add the 123 base pair band because it was very faint, but I assure you that it's there. And in lane 1, we have our 2 other bands that we expected with the wild type plasmid, and then our huge chunks in trans 1 and trans 2. And I cannot stress to you how many failed gels my lab partner Victoria and I ran. So the only proper meme I thought to express how happy we were was, "We did it, Joe. We got a successful plasmid and a successful gel run." Our next step was to isolate the YTK74 plasmid, which has the patch that will repair the double-strand break whenever we eventually introduce this CRISPR-Cas9 system into the yeast. And we amplified this URA3 patch with a PCR, or a polymerase chain reaction. This basically takes a chunk of DNA and makes a bunch of copies of it. And our amplicon of the PCR reaction was about 1,270 base pairs. And we ran this again on a gel, and we have our ladder of base pairs that we know how big those chunks of DNA are. Lanes 1 and 2 have our 2 replicates, and we were very happy because this was exactly the size of DNA we were expecting. However, lane 3 had a negative control, and we were expecting to see no band in there at all. However, we thought it was okay because there just might have been some contamination, and we decided to just go on with the experiment. Our next step is to transform the modified L201 plasmid and the URA3 patch into yeast and then select for transformants. And so once again, a transformation is the yeast with extracellular DNA. They eat the DNA and then express whatever genes are encoded. And so once we do this, we hope that we have the L201 plasmid, which codes for the Cas9 protein. It has the guide RNAs, and it also has a leucine oxytrophy gene. Leucine is an essential amino acid, and if an organism does not make the amino acid itself, and if the media does not contain the amino acid, it is unable to grow. Similarly, the URA3 patch that we amplified with a PCR, that confers uracil oxytrophy, which is an RNA nucleotide, which again, if we're not able to make it, or if there's no uracil in the media, it will die. And so we use leucine and uracil dropout plates. Each of these do not contain either leucine or uracil, and the leucine dropout plate was a control to see if the transformation actually worked. And then the uracil dropout plate was to see if the CRISPR-Cas9 system worked. The idea, again, was that if the yeast contained the modified L201 plasmid, it has that leucine oxytrophy gene and therefore able to grow. And if it has the patch that repaired the DNA after the double strand break by Cas9, it will be able to grow. And if it doesn't, it will be able to die. And on the very first try of the experiment, we got great results, which again, never happens. On the left is our uracil dropout plate, and on the right is our leucine dropout plate, and we have really great growth. And right now we're all celebrating, and our yeast are all celebrating too, because we have gotten great results so far. And what's next? Next is phenotype testing to see if we actually did increase pectinase expression. We were actually supposed to do this earlier this week, but the easiest step of our experiments are making media, and our auger did not solidify, so I don't know what happened. So come back to me next week, and I will have results for you. But we are going to use polygalactronic acid, or PGA plates, to see if this organism is able to degrade pectin. So these plates have pectin in the media, and what we do is we flood this media with iodine. Iodine is very dark, and it can bind to the pectin and stain it very dark. And if anything is able to degrade pectin, we will expect a light zone of clearing, and that tells us that whatever we put there is able to break down pectin. This upper left quadrant, we have an actual pectinase enzyme that was able to degrade the pectin. This bottom left quadrant is ester BCA 09448, which is the organism that was industrially weak but expresses a pectinase. And we can see there's degradation. This bottom quadrant here is ATCC, and as we expected, has no pectinase expression. This bottom right has bacteria that's capable of degrading pectin. This top right is water, which was a negative control. And after introducing the CRISPR-Cas9 into the yeast, we hope to see a zone of clearing for ATCC. And then after that, we will all be rich because we would have fixed global warming. I'm just kidding, but as a reminder, petroleum is a very large contributor to greenhouse gas emissions, especially in the transportation sector in the United States. And bioethanol is a very promising alternative, especially whenever using second-generation biofuels. And using yeast to make bioethanol will lead to lower costs of industrial bioethanol production, higher efficiency, as well as a higher ethanol yield. And before I end, I just have to shout out some people who made this possible. On the left is my mentor, Dr. Claire Edwards, and Emeka and Caitlin were in the Edwards 2024 lab, but they did not do this project with me. Derek was actually my lab partner, but I have no pictures of him anywhere. My middle picture is Victoria, who is my bestie, and she did all the CRISPR experiments with me. And then on the right is everyone who is in the Bainham and Edwards microbiology lab for the summer of 2025 and made doing summer research a blast. Thank you all so much.
Excellent job, Hannah. I'm so excited to hear your smart questions before the dumb questions. Audience questions.
Yes, Dr.
Yeah,
Bainham.
great.
Back to dropout plates.
Yes.
Those aren't replica plates.
Rupture.
They're not.
No.
Take that and then take the same ones and place them there.
No.
Is it possible that they could have plasmid but not the patch?
Well, we took just like 100 microliters from our transformation product and plated them onto each of the, so each transformation product grew on leucine and grew on uracil, and it's the same, like.
Each transformation reaction had bacteria, but not necessarily every yeast, that had, but not necessarily every yeast got both.
I guess, do you know that those are clonal?
No, we don't, but we are patching them onto leucine and uracil plates, and they're growing right now, so we're making sure that they have leucine and they have uracil.
You're doing that part now.
Yes, but they wouldn't be able to grow on the uracil unless the URA3 patch was actually in the backbone of the DNA, because then.
Oh, so the uracil patch is on the plasmid as well.
Well, it gets, like, whenever the double-stranded DNA break is induced by the Cas9, which is on the plasmid, and then we add the URA3 patch, if the patch is not within the DNA, then the proteases within the cell will degrade.
So the Cas9 is expressed as soon as the plasmid goes in. You don't have to, like,
Yes.
activate it. So it's presumably the patch is in the genome.
Yes, and then we're also going to do a gel that we have primers to look on either side of where the break should have been, and then compare the DNA sizes between the wild type and then the modified yeast. Yes, Dr. Stephenson.
You don't have to laugh like that.
Sorry.
Can you explain to me what a strain is?
Oh my gosh. A strain is all of the same species, but again, in evolution we learned that there is no such thing as a good definition of a species. But it's basically all of the same species, but they might express different phenotypes and different genes.
I had to do it, I'm sorry.
It's okay. You asked me the same question in class.
I have one, okay, my really dumb question is next, but, so is what I think what Dr. Greer was asking is like, why was it okay to do separate, like screen out the ones that don't have uracil and screen out the ones that don't have leucine, and choose this, like, why do you not have to make sure that?
Well, ideally, if they grow on the uracil, that means that that patch was introduced into the double-stranded DNA break that was due to the L201 plasmid, and that plasmid has the leucine oxytrophy chain.
So it should isolate.
The only way they could be redundant. Right?
Yeah.
The only way they could grow on uracil is if they could also grow on leucine.
Okay.
But not-
Doing
And
this
then,
is not show that. But
okay, then my really stupid question that I'm dying to know is where does all your clip art come from?
Oh, the what?
Like the clip art and the pictures.
Oh, there's this app or this website called BioRender,
Okay.
and I recently bought BioRender Plus, which allows me to really go crazy with all the pictures. Thank you, Tammy, for her credit card.
Other questions?
Yes, Dr. Stephenson.
So most of our biofuels traditionally have come from like, I forget what it's called, corn smut, if that's correct? So does corn smut have high amounts of pectin in it, like some of these other products that you're talking about, in terms of the viability of using it as like an industrial bioethanol producer?
I don't know if it has a lot of pectin in it, but I do know that because like corn competes with the food industry, the reason that we're looking to second generation is because like it's food waste already, so it's not directly competing with the food industry.
the corn that's used for bioethanol cannot be used for food. That's what you mean.
I think.
Yeah, that's correct. It says 96% of our corn is for not food. No.
Sounds like bioethanol.
Okay. Amazing.
I'm doing this like the current generation. I was going to print it out, and I was like, I guess I can just read it off my phone, but that requires me being able to see it. Here, I'm going to do
Your
this.
first time going to give, so this is Dr. Karen, sorry, who is going to present Hannah with her cord and stole. I think say a couple things on behalf of Dr. Edwards, who is in the conference. But I just want to say, so this is the cord and stole that Hannah will wear at graduation, and it'll represent not only the great work that she did on this project and her excellent use of bio-specific clip art, but everything she's done as an honor student, her amazing contribution to the St. community. We've been really happy to have pleasure to pass this over to Dr. Gehring, who will
Okay, so Dr. Edwards sent me a paragraph to read, and then I can't resist putting in my own 2 cents, but I'll read hers first. I'm very disappointed that I was not able to attend Hannah's honors thesis today, but I have no doubt that she excelled, as she always does. I first met Hannah in my human microbiome class in January 2024. Less than a month later, I emailed her to ask if she wanted to join my lab for summer research. Hannah has since taken ownership of her research in a way that few students do. With the help of her lab partner, Victoria, she established the CRISPR protocol she presented today. I had never conducted these experiments before, so Hannah truly is a lab expert. I just put in the orders and checked in periodically to see if I could help. She did this all while excelling in her classes, training for marathons, and getting accepted into medical school. I'm grateful for the opportunity to be part of Hannah's journey, and I look forward to seeing what she accomplishes in the future. Congratulations.
Yeah, thank you.
Thank you. I have a similar story. Hannah took molecular genetics. When was that? Fall 23?
Fall 2020. Oh, yeah.
Or was it
yeah.
fall 23, right?
Yeah.
And I don't know, halfway through we realized we were going to need another supplemental instructor for molecular, and I was like, I know who could be good at that. And unfortunately I only got to work with her for a short time because she decided she was too busy and had to focus on other things, which just tells you how mature she is because she can self-regulate and know where her balance is. So I was really lucky to get her for a short period of time. But she's amazing, and in every class I've had her in, I would echo everything Dr. Edwards said. So congratulations. Give me a word. And here is your soul.
Thank you.
Yay, congrats.
Thank you.
Good job.
Thank you.